Hello, students. I hope you are all seated for this class. All right, thank you. Today, we are going to look at a very interesting topic in biology, which is genetics. Is a very interesting and complex topic in biology. Now, what is genetics? Genetics in biology is the study of hereditary and variation. All right, that is the scientific study of hereditary and variation. Okay, what is hereditary? Hereditary on the board is the transfer of traits from the parent to the offspring without undergoing any disturbance. All right, some characteristics or traits are transferred from the parent to the offspring. All right. Here, variation is the sum of total differences which exist among organisms of the same species. All right. Sum of total differences that exist among organisms of the same species. And this variation here is referred to as genetic variation because they are linked with the gene. And genetics is the study of hereditary and variation. All right. What type of variation? We have genetic variations. Okay. We equally have environmental variation, and this is also studied in that is genetics on the board. Now, what causes variation in biology? Remember, during the cell division known as meiosis, all right, at the late prophase, at the late prophase. I've dropped this video in this channel. At the late prophase, there is exchange of genetic materials between bivalent chromosome. And this exchange of genetic materials between bivalent chromosome will now result to mutation. Mutation is sudden change in the gene, right? Mutation, mutation, got a spelling here. Mutation is changes in the gene or chromosome. Changes in the gene or chromosome. That is mutation. All right. Exchange of genetic materials causes mutation, and the mutation lead to genetic variation. You understand? Exchange of genetic materials causes mutation, which result to genetic variation. This mutation here that is caused by exchange of genetic material is also caused by sunlight, or you can say ultraviolet rays, right? It is caused by sunlight or ultraviolet rays. Okay, now we are going to look at genetic terminology. Genetic terminology. We are going to look at different genetic terms. Number one is genotype. All right? Genotype here is the genetic makeup of an organism or individual. That is the genetic makeup of an organism or individual. Okay? That is it on the board. Then number two, we have phenotype, which is the physical appearance of an organism. Okay? The number three is allele. Allele is a pair of a gene. A pair of a gene is allele. The number four, we're going to look at dominant. Number four, we're going to look at homozygous. Homozygous gene. All right? Homozygous gene here is the gene with identical allele. That is the gene of identical allele. If you have this kind of gene here, capital T, small t, this is an allele and this is another allele. All right? Now, when the alleles are the same for the case of this, like this, all right, it becomes homozygous gene because the alleles are the same. Here it becomes homozygous because the alleles are also the same. The number five, we have heterozygous gene. All right, this is the gene of non-identical allele. If you have a gene like this, AB, as the gene, alleles are not the same. All right, gene like AS, alleles are not the same. I mean, it has different alleles. Then gene like this, the alleles are never the same. We call that type of gene with different alleles as heterozygous gene. Thank you. The number six, we have dominant gene. All right, dominant gene is the gene that expresses itself in the presence of another gene. All right, that is the gene that expresses itself in the presence of another gene. The number seven is recessive gene. All right, that is the weak gene that did not express itself in the presence of another gene. All right, that is the gene that did not express itself in the presence of another gene. We call it recessive gene or suppressed. You can say it is suppressed gene or weak gene. They did not express itself in the presence of another gene. Okay, then the next we are going to look on the board now is going to be hybrid. All right, hybrid is the offspring of two parents of different species. Okay, the offspring produced by two parents of different species, both parents must be closely related. All right, that way is hybrid. All these are the genetic terminology. Now we are going to look at the experiment of the father of genetics. All right, the father of genetics. Okay, that is, yes, Gregor Mendel, Gregor Johann Mendel. You know that the gene that carries the family traits are located in the DNA. And every organism must possess DNA which contains the family traits or family characteristics. And that is why DNA test, DNA test is used to determine the paternity of a child. Okay, the test used to determine the paternity of a child is DNA test, and reason because DNA contains the family traits or characteristics which must be transferred from the parents to the offspring. All right, it is equally used to determine the maternity of a child. All right, just that we don't normally do maternity tests because once a woman is pregnant, it is always pregnant and it's obvious. Everybody knows that she is pregnant, but you don't know the father, the man responsible for the pregnancy. That's why we constantly do paternity tests to confirm the father of the child. All right, then this DNA here is also seen in chromosome. All right, because chromosome is formed by condensation of the chromatin thread, nucleolus, and the nucleoplasm, all located inside the nucleus. If here is the cell, for example, we have the nucleus, we have the cytoplasm here. Now, the study of genetics is concentrated in the nucleus. Okay? Now, inside the nucleus of the cell, we have the nucleolus, we have the chromatin thread. Okay, then here we have the nucleoplasm. Now, this is chromatin thread, right? This is nucleolus, right? Then this is nucleoplasm or nucleoplasma, okay? Now, I'm not going to draw all the components we have inside the cell. We're going to concentrate only on the nucleus of the cell, all right? During the formation of chromosome, the nucleoplasm, the chromatin thread, and the nucleolus condense together to form chromosome. And the nucleolus is the structure that houses the DNA inside the nucleus of the cell. And since nucleolus here condenses with nucleoplasm and the chromatin thread to form this, that also shows that the chromosome contains the DNA which carry the family traits, right? And that is why you can say that chromosome contains the genetic or hereditary materials, which I described on the board as family traits. All right. Now, the structure of chromosome was discovered by a man called Morgan. Morgan discovered the structure of chromosome, all right, using a fly known as fruit fly. Right? Now, what structure of the fruit fly did Morgan use to discover the structure of chromosome? He used the salivary gland of the fruit fly. Do you understand? Morgan uses the salivary gland of the fruit fly. Now, in your past question, the spelling of fruit fly is very important. What is the name? The spelling of fruit fly is Drosophila melanogaster. Drosophila melanogaster. That is the scientific name of fruit fly, which Morgan used its salivary gland to discover the structure of chromosome. Then in your exam question, you were asked which of the following organism is extensively used in the study of hereditary theory. The answer is Drosophila melanogaster, which is fruit fly. You have other organisms like Musca domestica. Musca domestica is house fly. We have another option as Z maize, which is normal maize, right? Then we have Homo sapiens, man, right? Then now listen, reason why Morgan uses the salivary gland of fruit fly to discover the chromosome is because the chromosome of the salivary gland of the fruit fly is very large in size, right? It's larger than the chromosome of man. And that enables him to view the structure, the component of the chromosome. Okay? That is why Morgan used fruit fly on his experiment. Now, let's now look at the father of genetics, the experiment he did, Gregor Mendel, the father of genetics. Let's find out what he did here in the study of genetics. Gregor Mendel here, in his experiment, used pea plants. Right? He's trying to discover how traits are being transferred. Okay? He used pure tall and pure short. Right? The pure tall here is homozygous tall. Then the pure short here is homozygous short. Do you understand? Now, why did Gregor Mendel use pea plants on his experiment? He used pea plants because pea plants complete their life cycle within 1 year. All right? Complete the life cycle within 1 year to enable him to see the result of his experiment within a short period of time, which is the 1 year life cycle. Then he used pea plant because pea plant can undergo both self and cross pollination. Both self and cross pollination. Now, when I say pure tall here, what Gregor Mendel means by pure tall is that he took a very tall plant and according to his experimental work, he shows capital T and capital T as the gene for tall, then small t and small t as the gene for short. All right? Gregor Mendel here cross this against this, right? And this against this. This gives capital T and small t. This gives capital T and small t. Then this against this gives capital T and small t. This against this gives capital T and small t. We call this the first filial generation. Okay? Now, we observe that all of the first filial generations are tall, and this got him confused. He was trying to react like how, why? We have this gene for short, all right? I crossbreeded between short and tall, and all of the first filial generation, which is the offspring, all of them are tall. Does that mean that gene for shortness is not transferred to this plant? All right? He have to prolong, like to extend the experiment to find out the final result to conclude his experiment. All right? He now allowed the plant to undergo self-pollination. All right? But look at the experiment that illustrates what he did here on the second experiment by allowing the plant to undergo self-pollination. Now, if this is an offspring now to undergo self-pollination, you know that both the male and female gametes of the same plant must have the same gene, right? So he now showed the second experiment here between this and this, right? Then he crossed this against this, cross this against this, having capital T and small t, cross with this, capital T and small t, cross with this, capital T, small t and small t. All right? And he called this second okay, he said that this is the second filial generation. All right, this is capital T and capital T. We observe that 1, 2, and 3 are tall, all right, but in terms of genetic makeup, all right, let's say genotypic ratio now. In terms of gene, this is homozygous tall, this is heterozygous tall. heterozygous tall, homozygous short. All right. We observe that this gene here is 1 because it is only one among the 4. Then here, these guys here are 2, and this one is single 1. Instead that the genotypic ratio is 1:2:1. Then here, genotypic ratio here is 4:0 because all of them are the same gene. All right, this is genotypic ratio. Then in terms of phenotypic ratio, let me use PR, phenotypic ratio. We observe that 1, 2, 3 are all tall, all right? Then only one is short. Okay, now state that, oh, this gene here that are all tall, they have another gene for the short, but that gene did not express itself. And this is what led him to discover the theory of dominance, that some genes dominate other genes. All right. So it says that in the second experiment, 3 of the second fillet generations are tall and 1 is short, showing that the short gene from the grandparent is transferred to the first fillet generation, which later becomes dominant here out of the 4 offspring of the second fillet generation. All right. Then it says that the probability of this organism to produce 3 offspring that are tall is 75%. The probability of producing tall offspring is 75%. Then probability of producing short offspring here is 25%. Then for the genotypic ratio here, this is 25% for this gene, this is 50% for this gene, this is 25% for this gene. All right, and here Gregor Mendel said that the first gene of the heterozygous is the gene for dominance. The first gene of the heterozygous is the gene for dominance, while the second gene of the heterozygous is the recessive gene. And this will help you to answer a lot of questions in your exam. Okay, that is all about the Gregor Mendel experiment. Okay, is there anything I've not explained? All right, I will stop here. In the next video, we are going to look at different exam questions, especially on this topic in the next class. Please don't forget to subscribe to our channel. Thank you and God bless you.